Cognizant to the needs of our learners, parents, and teachers, the Department of Education provided us with powerful tools for productivity that will allow us to foster critical thinking skills, problem-solving skills, communication and collaboration skills to be compassionate, responsible global citizens. Join us and discover new ideas in our series of professional development training program with the ICTS Edtech Unit and Microsoft Education Philippines. Together, we will equip our learners and empower our fellow educators for a dynamic future. Para sa bata, para sa bayan, at para sa guro. Sulong Edukalidad.
Magandang araw, Sir Wilbur po, at your service. Narito ang ETUlay upang gabayan ka sa inyong pag-aaral upang lubos na maunawaan ang iba't ibang paksa o subject. Ang ETUlay ay isang free online tutorial class na pinangungunahan ng ICTS Educational Technology Unit sa pamumuno ni Undersecretary Elaine Del B. Pascua. Ang programang ito ay hindi lamang para sa mga bata, kundi ito rin ay magsisilbing gabay sa mga magulang at mga guro kung paano nila ituturo o gagabayan sa bawat asignatura ang kanilang mga anak o mga estudyante. Sa kasalukuyan, ang self-learning module mula sa rehiyon ng Calabarzon at kilala sa tawag na pivot ang ginagamit sa ating ETUlay online class. Kaya ano pang hinihintay ninyo? Ihanda na ang inyong mga ballpen o lapis, papel o kwaderno at samahan kaming itulay ang pagkatuto para sa bawat batang Pilipino. Sama-sama tayong magtutulungan para malampasan ang mga hamon sa panahong ito. Halina't matuto kasama ang inyong online tutor sa oras na ito.
Alright, it's a happy Monday mga ka-ELS. So good afternoon po sa ating mga live viewers who are viewing live from Facebook and YouTube. So today we're going to have our session for senior high school, Earth and Life Science. So we are now on quarter 4, week number 4. At ang ating focus for this particular session is alamin natin ang mga konsepto tungkol sa genetic engineering. Alright, so sa mga bagong viewers po natin dyan, maybe you're a parent, you're a teacher like me, or you're a student, a grade 11 or grade 12 student taking up senior high school Earth and Life Science. So my name is Teacher Tony or your tutor Tony for this afternoon. So samahan niyo ako sa susunod na 40 minutes ng pagtalakay at pag-appreciate sa ganda ng ating mundo at ng buhay na meron ito. Alright? So, as mentioned sa ating teaser kanina, no, so we are using the module provided by Region 4A Calabarzon or the Pivot module. For this session, we will be using two modules. Module number 24 entitled Genetic Engineering, and of course, another module, module number 25, entitled Benefits of Genetically Modified Organisms. All right, so are you all ready? Ayan. So I hope ready na ang inyong mga papel or lapis or ballpen or any writing materials with you. Kung meron kayong kopya ng module, mas mainam. And of course, kailangan din ng presence ng heart at saka presence go on our discussion. Comment nyo lang inyong mga pabate, mga shout-outs. So don't forget to mention the name of your school and location kung saan kayo nanonood right now. Alright, so I hope everyone's ready. So I hope you're starting your week right. Ayan. So actually, ayan, so we are now in the month of June and alam nyo ba, so maikling patalastas lang before our discussion. Ano? So June is the environment month here in the Philippines. So last Saturday is, June 5, we celebrated World Environment Day. At bukas naman, June 8, we will be celebrating World Oceans Day. So back-to-back yung mga celebrations natin because June nga is the Philippines is celebrating the Environment Month. At ang theme natin for this year is sama-samang pagkilos, sama-samang paghilom. Ikaw at ako, tayo ang kalikasan. So napakagandang tema, very timely. No? So sana sama-sama nga tayong kumilos para sa sama-samang paghilom ng ating planetang Earth. Alright? So yan. just to refresh your minds, no. So last session we had our session for perpetuation of life. So specifically, we discussed about asexual reproduction at saka sexual reproduction ng mga representative organisms sa animal kingdom. Alright, so for this week, itong ating mga session objectives. Ayan, so first we have to describe the process of genetic engineering. So naalala niyo pa ba yung konsepto ng genetic engineering? And of course, we have to evaluate later on the benefits. Ayan, evaluate the benefits and risks of using genetically modified organisms or GMO. So I'm sure narinig nyo na yung mga GMO na yan. So re-refresh lang natin yung minds nyo. And then later on, we'll weigh the risk and the benefits na binibigay ng mga genetically modified organisms. I hope you're all set. So let's begin. So before we proceed talaga with the discussion of genetic engineering, refresh lang natin ang inyong mga minds about genetics. Kasi we're talking about genetics. So ano nga ba ang genetics? Genetics is the branch of biology concerned with the study of the DNA. I hope you still remember what is DNA. Deoxyribonucleic acid, the molecules of life. Ayan. So how their DNA manifests as genes and how those genes are inherited by offspring. So madaling salita, so sa genetics, pinag-aaralan natin ang heredity at saka variation. So heredity means the passing of traits from the parents tas mga magiging offspring niya in the generations to come. And variation kung paano nagkakaiba-iba yung mga living organisms because there's a variety or individuality ng mga genes natin. Alright? So that is genetics. So in the study of genetics, of course, may encounter natin yung words na chromosome, DNA, at saka genes. Ayan. So if you're going to have a sample illustration for this, so we all know that, yan, so start tayo sa chromosome. So ano nga ba yung chromosome? So nalilito kasi iba sa chromosome, sa DNA, at sa gene. So padaliin lang natin, no? So inside the nucleus of the cell, sa dito, ang pinakita is the human cell. So we can find the chromosomes. So ano nga ba yung chromosomes? The chromosomes are thread-like structures located inside the nucleus of animal and plant cells. So each chromosome is made up of protein and a single molecule of deoxyribonucleic acid or yung tinatawag natin na DNA. So para siyang compare natin siya sa isang yung pinatatahin na notebook, yung yarn, ayan. So the yarn represents the chromosome. Tapos kapag inentanggal mo, pag kinuha mo yung isang piece nun, that will be your DNA or the deoxyribonucleic acid. So sinabi ko nga kanina, the DNA is the molecule of life. So ito yung pinapasa from the parents to the offspring and it contains the specific instructions that make each type of living creature unique. So particularly, yung mga meron tayong iba't ibang klase ng DNA and of course, iba't ibang klase ng genes. It carries the instructions for assembling the proteins kung ano yung magpamanifest sa ating phenotype. So mamaya, recall natin din yung terms na genotype at saka phenotype. So yun na. So we have the chromosomes. Ayan. So inside the chromosomes, the thread-like structures, ayan, makikita natin ang DNA, the double helix structure. Ayan. So it's a nucleotide. And of course, yung bawat segment or bawat particular portion ng DNA ayun, is responsible for a particular trait. Ang tawag doon ay genes. Okay? So chromosome, DNA, and then yung segment or bawat particular or distinct portion ng DNA is what we call the genes. Alright? So the genes, these are the coded instructions for everything that must happen in the body. So how we function and how we look. Say for example, there's a gene for the eyes. Anong kulay ng eyes natin? Black ba yan? Brown or blue? So there's a particular gene din sa katawan natin na magdidictate kung ano magiging appearance ng ating hair. Say for example, so do you have a straight hair, curly hair, or a wavy hair like me? Ayan. So may mga particular genes. So may genes din tayo for height. Diba? So may mga ganon. Alright? So genes will dictate the particular phenotype. Alright? So chromosomes, DNA, and then the DNA contains specific portions called the genes. I hope naintindihan yung differences between the three terms. Alright. We also be using the terms genotype and phenotype. So madali lang naman to. So pag genotype, we'll be dealing with the genetic makeup of the individual. So the DNA siya sa genes on the molecular level. Pag phenotype naman, ito yung minamanifest ng anong klaseng genes meron ng isang individual. Kumbaga, yung morphology niya or yung physical appearance. So in our slide, phenotypes are the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. So genotype, the genes, phenotype is the physical appearance or the morphology of the organism. Ayan, so good afternoon sa ating unang commentator. Yes, commentator. Parang nanonood ito dati. So Heavenly Heart, Tanghian. Ayan, good afternoon sa iyo. Taga saan ka ba? Mention mo naman. And I hope sa mga nanonood po, please like and share our session para at least ma-reach out din natin yung mga iba pa nating mga viewers, especially our senior high school learners. So ano nga ba yung genetic engineering? So this one in our diagram or representation. So in the middle, we have the DNA, ayan, yung double helix structure niya. And we have here a geneticist or perhaps a molecular biologist. And as you can see, ginagalaw na yung bawat mga pairs or mga particular genes dun sa DNA. So what is genetic engineering ba? So genetic engineering is the artificial manipulation. So ibig sabihin, ginagalaw yan. Modification, binabago, and recombination of DNA. So mamaya kasi, one technique sa genetic engineering is kumukuha ng gene sa particular organism tapos ini-insert sa isa pang organism. So ganun yung magic or yung process ng genetic engineering. So or other nucleic acid molecules in order to, ang goal kasi is to modify an organism or population of organisms. So sa biology kasi, meron tayong mga risk nga at saka benefits ng genetic engineering. So I hope in our session ma-highlight yung benefits, ma-highlight yung benefits ng different genetic engineering techniques. All right? So that is the definition of genetic engineering. Ayan. So isa-isahin na natin yung mga i-discuss natin na different genetic engineering techniques. So we have under artificial selection, meron tayong tatlo. We have selective breeding, hybridization, at saka inbreeding. So, di-discuss natin yan isa-isa later on. We also have the techniques called cloning. Ayan, yung mga clones. I-mention ko na yung last sessions, di ba? Cloning. We also have gene splicing. Ayan, ini-splice, kinakat ang genes. Gel electrophoresis. Ano kaya yung gel electrophoresis na yan? At saka yung very common or very famous na technique, we have the recombinant DNA technology that is being used in different areas or in different fields in sciences. All right? So those are the techniques. Isa-isahin na natin. Ayan. So we have the first technique called selective breeding. So from the word itself, you select a particular trait. Siyempre yung trait na yun ay dapat desirable. Ibig sabihin magandang klase ng trait. And you breed. So pinag-breed mo yung particular organisms. All right? One of the best examples sa animal kingdom is, ayan, yung dogs. We have different kinds of dogs. Ako kasi, I'm not so particular with or I'm not familiar with the different breeds of dogs, no? Ang alam ko lang yung Aspin, yung common sa Pilipinas. Ayan. Although, marami tayong mga fur parents, di ba? And also mga fur parents natin dyan, they have different kinds or different breeds ng mga dogs, di ba? So, paano nga ba nagkaroon ng mga different breeds ng dogs? So, yan ay produkto ng tinatawag natin na selective breeding. Breeding is also known as artificial selection. This is a process used by humans to develop new organisms with desirable characteristics. So yung mga dogs, di ba, particularly, they are after the intelligence, the size minsan, or depende sa location kung ano yung magiging tulong niya for his or her magiging companion or amo. Alright? So ang ginagawa ng mga breeders, they select two parents that have beneficial phenotypic traits, yung phenotype, yung appearance, to reproduce. And then they will yield or they reproduce offspring with those desired traits. Ayan. So tingnan lang natin, trace lang natin yung history, no? Ayan, so sinasabing as early as 14,000, at least 14,000 years ago, nagbe-breed na ng dogs, yung mga dog breeders. And ayun nga, ang sabi, mga dogs ay nanggaling or the dogs that we know right now, evolved from the wild gray wolf or the Canis lupus. And because of the different selective breeding techniques na ginawa ng mga tao all around the world. So we have Europe, America, China, India. So here are some of the key locations. They have successfully breed different types of or the different breeds of dogs. Ang target syempre dyan is ma-highlight yung size nila. Kailangan ba malaki, medium size, or maliliit, mga cute for pets, di ba? Or yung intelligence for certain tasks. Although we know that dogs are really intelligent animals and they are great companions for hunting, pag-shepherd ng mga other animals. Ayan. So yun yung one of the best examples of selective breeding in animals. So sa plants naman, meron din tayo. Sinong kumakain dito ng, ayan, broccoli? Ayan. One of my favorites, very rich in protein and other vitamins and minerals. So broccoli, did you know that broccoli is man-made? So technically, wala talagang broccoli, no? It's man-made. So paano nangyari yon? Dahil sa selective breeding din ng mga plants. It was made over 2,000 years ago by farmers, selectively breeding different types of cabbage or the wild cabbage. So ito yun, tignan natin yung magkakamag-anak. So na-mention natin si broccoli. Ayan, yung broccoli, yung cabbage, the turnips, the kale. Ayan, sarap yan sa salad. We have the cauliflower and the kohlrabi. I'm not familiar with that. Pero I'm sure kinakain din yan or pinapropagate din yan, pinaparami sa ibang areas ng mundo. Yan. So anong ginawa ng mga breeders, the farmers? By isolating wild cabbage or the wild mustard with specific characteristics, they were able to create a variety of vegetables. So from a single type of or a variety of vegetable, they were able to produce around 6. So ganun ka important, no? Kung imagine kung hindi yan na ginawa ng mga early farmers. So wala, hindi natin ma-enjoy kung yung broccoli, yung cabbage, and mga favorite natin, cauliflower sa ating chop suey or sa any vegetable dish or sa mga mahilig dyan sa salads, di ba? So that is a product of selective breeding. We also have another staple food, yan, bukod sa rice dito sa Pilipinas, sa mga agricultural countries, one of the staple food is corn or the maize. Alam niyo ba na ang corn ay nanggaling sa isang smaller plant na nagpo-produce ng kernels called the teosinte. The teosinte, ayan. Ayan. So paano nangyari yon? It was also selectively bred. Ayan. So from smaller kernels, yan, pinalaki siya ng pinalaki. Kinuha yung mga species or varieties that can produce bigger kernels. So, ang sabi dito sa facts natin, scientists believe that early Mexican farmers selected only the largest and the tastiest. Or remember, hindi lang sa size ng kernel, pati sa lasa din, of course, it will also matter. The tastiest kernels of the teosinte for planting, rejecting the pernier or the smaller kernels. This process allowed the Mexicans, so mga Mexicans pala or sa Mexico pala nag-start, no, to develop corn very quickly. So and then the rest is history. And actually, we have different varieties na rin ng horns, di ba? We have the sweet corn, the Japanese corn, yung corn na puti, yung mga native na corns natin sa Central Luzon, di ba? Ayan. So that is also a product of selective breeding. Ayan. Ayan. So we have selective breeding first. Okay. So proceeding with the second genetic engineering technique called hybridization. So, keyword naman dito is hybrid. So, pag sinabi kasi nating hybrid, it's a product of two individuals with unlike characteristics. Magkaibang characteristics na they are crossed, they are breed to produce the best in both organisms. Ma-highlight syempre yung magandang characteristics or phenotypic structure niya. So, one great example for hybridization or yung hybrid is yung ginawa ng isang American plant breeder and horticulturist, so nag-aalaga, nagpa-propagate ng mga plants, na si Luther Burbank. So, isa siyang American. So, nagkaroon kasi ng famine, nag-i-infest kasi or na-infect yung mga potatoes during his time. If you would recall, baka talop or nakapagbalat na kayo ng potatoes before, parang si Princess Sarah. Minsan, may mga patatas tayo natatalupan na meron siya mga black na di ba? So that's actually infected by a certain type of fungus. Ayaw natin ng ganun, okay? So ang ginawa ni Luther Burbank during the time na sobrang lumaganap yung infections sa mga potatoes, what he did was, he crossed a disease-resistant plant. So sinabi natin disease-resistant, a plant or an organism that can resist any type of disease. Alright? Tapos kinaros na yon sa isa pang type ng potato na nakakapag-produce ng maramihan. So yung mga produce niya or yung yields niya ay mga malalaking kinds of potatoes. Alright? So na-produce ang ating mga Burbank potatoes. Ayan. So thanks to Luther Burbank, Ayan. So na-survive nila yung famine na yun or yung infestation ng mga fungus sa potatoes. So that's one example of hybridization in plants. Let us now proceed naman with, ayan, is an example of hybrid naman sa mga animals. I'm sure you have heard of the word liger. Ayan. So liger, ito, picture na to kasi kinuha ko sa website ng National Geographic. So we have here a liger. Ang name niya is, ito yung mother na liger. A liger, Zita, walks with her month-old cub at Russia's Novosibirsk Zoo. And ligers are the offspring of a female tiger, kinag-cross ang female tiger at saka male lion. Alright? So, ayan. So we have here a liger, a hybrid. Other hybrids din, meron tayo tinatawag na tigon naman. So it's a product of a female lion and a male tiger na pinag-breed or pinag-cross. We also have a leopon, a combination naman ng lion at saka leopard, at saka isa pa, jagulep. Jagulep is a combination of jaguar and leopard. So bakit sila pwedeng i-breed? Kasi they are from a common ancestor. So they are a family of cats. So their genes can be crossed. Ayan. So, liger, tigon, leopon, at saka jagulep. So, examples of hybrids and animals. And of course, di tayo pahuhuli sa Pilipinas. Medyo matagal nga lang to, 1962. Ayan. So, a zebrunky, a rare animal that is half zebra, so half zebra naman, tsaka half donkey. So, related sila, no, yung mga kabayo na yan. It was first bred in Manila Zoo. Ayan. So, sana mag-open na yung Manila Zoo para madala ko ni anak ko. Manila Zoo in 1962, and they named her Lolita. So Lolita, the zebrunky sa Manila Zoo. It was bred in 1962. Ayan, so a little trivia for all of you. So those are the hybrids. And lastly, for artificial selection, we have a technique called inbreeding, wherein organisms that are genetically similar are bred to maintain desired traits. So ang nahanap natin na research dito at saka sa module found on module number 25, ayan. So we have what we call the Holstein dairy cattle. So ang cattle kasi, as you all know, they are, we rely on cattle or cows for their meat at saka milk. Ayan. So very, din yan, staple sa mga families natin, di ba? So for example, in breeding Holstein dairy cattle has led to increase of milk production and target, of course, makapag-produce ng mas maraming milk. Kasi nga, syempre, in terms of population, mas dumadami ang tao. So kailangan increase din yung production, di ba? So for food security purposes, pero meron siyang disadvantage. The cows are more difficult to breed once they were once they went the process of inbreeding. So yung mga next generation, mas nahihirapan na rin mag-breed. Alright, so that's one of the best examples of inbreeding. So once again, under artificial selection, we have inbreeding, selective breeding, at saka hybridization. Alright, let us now proceed to the more technical na mga techniques, mas elaborate na techniques because they are gumagamit na ng mga laboratory equipment, mas high-end na mga tools para makapag-produce ng mga organisms. Yeah, so the first one is called cloning. Cloning is a technique that scientists use to make the exact genetic copies of living things. So general kasi in term sa biology, yung clone is an exact copy of the parent organism. Ayan, so wherein the genes, the cells, or the tissues, and even the whole animals can all be cloned. And of course, ang pinaka-best example niyan is noong 1996, ayan, si Dolly, the first clone chip, and eventually, tinry nila kung makakapag-produce siya ng offspring, and successfully, she was able to give birth to her, ano man tawag sa baby na sheep, the lamb, ayan, na Bonnie, ayan. So successful ang cloning procedures, naging instant celebrity yan si Dolly. We also have other clone animals, so I'll be showing sa mga series ng slides natin later, makita niyo yung mga clone animals. Like here in Japan, so we have Noto and Kaga. These are the cows duplicated several times. So from dalawa, naging mas marami pa. To produce more milk at saka meat. But, that was 1998 in Japan. We also have other examples right here. Ayan, mga cute na pigs. piglets were cloned. 5 piglets named Millie, Alexis, Krista, Dorcom, and Carol were cloned by a company wanting to reproduce. Ayan, have you heard of the news na yung organs kasi na pwede nating may clone derived from the cloned pigs can be transplanted to humans. Just like yung mga important organs natin na kailangan for transplantation like liver at saka heart. Ayan. So they are trying to clone these pigs para makapag-produce ng mga human organs for transplantation. That was, ayan, during 2000s. We also have, ayan, a cute na monkey, a rhesus monkey named Tetra. It's being cloned by scientists for laboratory experiments naman during the 2000s din. We also have another example, ayan. Ang cute na si Sisi or si Carbon Copy. It was cloned by a company wanting to go into business naman. Ako, business venture for for reproducing pets during 2001. So tuloy-tuloy na yung yung cloning procedures natin. Syempre kung may pusa, may aso. Snuppy the dog was cloned by South Korean scientists to study for medical purposes. Snuppy was cloned to study human diseases. Ayan, it happened 2005 in South Korea. Ayan. So those are some examples of cloning in animals. Ayan. So that's that second technique, no? So we have the first technique. So we have cloning. The next technique is what we call the gene splicing. Pag-splice. Pag sinabi natin pag-splice, to cut, tapos remove natin on a particular area. So when we say gene splicing, as our diagram suggests, so we have here double helix structure na DNA, tapos yung gene ng particular segment, kinat, tapos inilagay sa isa pang portion naman ng DNA. Alright? So gene splicing is the process of chemically cutting DNA of one organism and it will be put into another organism using restriction enzyme. So mamaya discuss natin yung restriction enzyme. So yung nagka-cut ng particular segment ng genes. Ayan. So para smooth lang ang pag-cut tapos pag-replace ng mga particular gene segments natin. Okay? So because of gene splicing, scientists can add and tag sequence to track and study gene products in an organism on the molecular level. It can also create new gene sequences para makapag-create ng panibagong variety of proteins with multiple or entirely new functions. So ginagamit yan sa mga researches at saka sa pag-develop ng mga panibagong genetically modified organisms. The next technique is what we call gel electrophoresis. Ayan. So as you can see, ayan, meron tayong device dyan. So nakasaksak. So meron siyang certain amount of electricity para ma-charge. Ayan. So it's a laboratory method used to separate mixtures of DNA, RNA, or proteins according to molecular size and charge. So imagine that on that molecular level or na microscopic level na nahihimay ng mga geneticists natin, ng mga scientists, yung DNA, RNA, and mga proteins. So imagine, iba't ibang klase kasi ng proteins ang meron tayo sa katawan. So imagine this innovation called gel electrophoresis. So paano nga ba nag-work yan? So we have another diagram here. So we have the negative and the positive charge. Of course, it involves electricity. Tapos meron tayo dyan plate. Ayan. Tapos padadaanan ng certain amount of electricity. The DNA will move. Ayan. ACGT, adenine, cytosine, guanine, and thymine. And it will produce a certain photo. Ayan. Ayan. So we have here a gel, agaragose gel placed in this buffer-filled box, yung kanina sa diagram natin. It's being powered, positive tsaka negative terminal. And then, ang goal is mapamigrate, mapamove yung DNA towards the camera para makapag-produce ng clearer picture at saka clearer separation ng mga constituent parts ng DNA and other biomolecules, yan, protein nga, yung sinabi ni teacher kanina. Alright? So, gel electrophoresis has several uses. Ayan. So, first is to separate DNA fragments for DNA fingerprinting to investigate crime scenes. So, ginagamit yan sa mga pag-investigate ng mga crimes. To identify kung kaninong mga fingerprints yon or kung ano mang ebidensya, di ba? Next is to analyze results of polymerase chain reaction to to analyze genes associated with particular illness, pag-identify ng mga or pag-aaral ng mga different illnesses natin. Pag-profiling ng DNA para ma-identify yung mga ancestors. Yan, sa evolution naman to ginagamit. Tsaka sa taxonomy. It also being used in paternity testing using DNA fingerprinting. So para malaman, di ba, kung sino ang tatay. Ayan. In the study of structural, ayan, kanina, function of proteins, pag-analyze ng antibiotic resistance ng organism, and of course, in the study of evolutionary relationships by analyzing genetic similarity among populations or species. So hindi lang siya sa research, it's also being used in taxonomy and in the study of evolution. Yan. And then finally, we have the recombinant DNA technology. So ano nga ba yung recombinant DNA technology? Recombine. Ayan. So yung mga key concepts natin na dapat tandaan for this, this is the joining together of DNA molecules from two different species. This is used to remove and insert genetic sequences from and into other sequences of other organisms. And then later on, sa mga diagrams natin, maririnig nyo yung mga terms na restriction enzymes, vectors, at saka host organisms. And it's being used sa production ng gamot, mga health supplements, cosmetic products. Of course, we have chemicals and other products na very beneficial sa ating mga tao. Okay, let's begin. So just to give you a preview, ayan, so we have here a plasmid. So plasmids are produced by bacteria. So commonly, yung mga bacteria kasi yung ginagamit as vectors, no, para maparami ang particular, let's say for example, a hormone or an enzyme. So ang ginagawa yan sa plasmid, we have restriction enzymes. Yung foreign DNA na gusto nating paramihin. Ayan. So restriction enzymes are the ones who will cut a specific segment of the DNA. Ayan, tapos i-insert. And the ligase enzyme ang magsiseal naman. And then this, yung recombinant DNA na na-produce, ang mapaparami. I-insert yan sa bacteria para paramihin. Okay. Yan, plasmids are the vector. So when we say vector, it is a DNA sequence that can transport. So pwede siya natin i-transfer from one cell into another para makapag-replicate siya para maparami. So yung sinasabi naman nating restriction enzyme, commonly sa mga reference books or sa internet sources, mababasa ninyong ECOR1. Okay? Or the restriction enzyme na nagki-cleave or nagka-cut ng specific DNA na pwede nating tanggalin or i-remove. Tapos papalitan natin ng gusto nating DNA na paramihin. Okay? An example here is yung pag-introduce ng human growth hormone sa plasmid ng bacterial chromosome. Ayan. So the same process lang din. Ayan. It uses the restriction enzyme and plasmid, of course, as the vector. Tapos yung pinaka-host organism mo is the bacterial cell na magpaparami ng growth hormone. Okay? Based on my research, yung growth hormone na ito is hindi yung growth hormone na inisip natin nagpapatangkad. Yung growth hormone na ito ay ginagamit sa mga baka or sa mga cattle para ang target na trait dito is yung trait na makapagpaparami ng production ng milk. Alright? So that's the technique called the recombinant DNA technology. Ayan, so recall lang natin yung mga techniques that we have discussed. Under artificial selection, we have selective breeding, hybridization, at saka inbreeding. We also discussed about cloning, gene splicing. So cloning, you are providing the exact copy, di ba? Gagawa ka ng exact copy. Gene splicing, magka-cut, tapos transfer sa another segment ng DNA or ng gene. We also have gel electrophoresis para pag-analyze ng DNA. And then recombinant DNA technology. Okay, so we are now ready to answer Match Me on Module 24, page 11 to 12. So your choices are letter A, artificial selection, B, selective breeding, C, hybridization, D, inbreeding, E, cloning, so F, gene splicing, and G, gel electrophoresis. So technically, yung mga techniques na diniscuss natin. So number 1, animals with desired characteristics are mated to produce offspring with those desired traits. Anong tawag dito? An example nito is in Dachshund. Dachshund is a kind of dog. Dachshund were once bred to hunt badgers and other burrowing animals. Yung Dachshund, ganyan. Maliit, short na dog, tas pahaba siya. So, ang goal niya is para makapag-hunt daw ng mga badgers at saka ng iba pang burrowing animals sa soil. So, what technique is that? Okay, so the correct answer is B, selective breeding. Number 2, creating an organism that is an exact genetic copy of another. They will have the same exact DNA as the parent. Of course, exact copy, we have the, ayan, so cloning. Number 3, DNA is cut, ayan, yun yung clue natin na, clue word natin. DNA is cut out of one organism and then put into another organism. A trait will be transferred from one organism to another. That is your F, gene splicing. Alright, may sumasagot. Thanks to, ayan, Jeremiah Capule Gallardo. Hello. I think you answered correctly sa number 2, letter E, and then letter F. Ayan, good job. Jeremiah Capule Gallardo. Lagay mo naman kung anong school ka para ma-acknowledge natin. Okay? Let's try. Others, you can also interact with me. Answer kayo sa comment box. Huwag kayong mahiyang sumagot. We are now number 4. Remember kanina yung sa Burbank potato created by Luther Burbank. So he crossed a disease-resistant plant with one that had a large food-producing capacity. That is your hybridization. So a hybrid example natin yan sa plants. Number 5, a technique used to compare DNA from 2 or more organisms. That is your gel electrophoresis. Diba? Kino-compare yung DNA, RNA, at saka yung mga protein structures natin. Ayan, Jeremiah Capule Gallardo, biotechnology teacher here po from Bataan National High School. Lessons po namin yan. Hello po sir, good afternoon po. So I hope makatulong po itong session natin sa inyong mga estudyante. Alright, we also have number 6. Breeding of organisms that are genetically similar to maintain desired traits. Yung example natin dito kanina is yung cow, yung cattle. It keeps each breed unique from others. The correct answer would be inbreeding. So, inaano ko na, so pina-fast forward ko na medyo yung exercises natin to help you out and to maximize yung time natin. Kasi meron pa tayo i-discuss. Alright, so here now is the discussion about the benefits of genetic engineering, or as mentioned by Sir Gallardo. So it's a component of biotechnology where you combine or apply the concept of biology para makapag-produce ng mga genetically modified organisms for the benefit of humanity. Okay, so pag sinabi natin kasi GMO, iniisip ng mga tao is yung mga they are common with genetically modified organisms na particularly sa mga crops or sa food. So, meron tayong dyan rice, of course, yeast, papaya, we have canola oil, and other food products, yan, tomato, corn, ayan. And in the Philippines and other Southeast Asian countries, I don't know if you're familiar, we have what we call golden rice. It's a genetically modified rice. So, yellow siya kasi meron siyang component ng vitamin A. And based kasi sa studies, vitamin A affects or greatly affects yung mga children natin dito sa Southeast Asia. So, sa Philippines nga. So, medyo kinokontra lang siya ng mga different organizations. But based on my research, just recently, 2019, na-approved na siya for staple, for consumption. So, based on studies, it's just like the normal rice incorporated with vitamin A. Okay, so that is your golden rice. Ayan, for ano naman, so para makapagbigay pa ako ng other examples ng GMO, ginamit ko yung module number 25, page 11, the exercises about transgenic or genetically modified organisms. So we will be having a guessing game. Sana may mag-comment. Ayan, alright. So magpapakita lang ako ng description tsaka picture just like this. Then hulaan nyo kung ano yung tawag. Okay? First example is an apple that tastes like grapes. Tingin na ba kayo ng ganyan? So meron yan sa US kasi. I haven't tasted one. So ang tawag dyan ay grapple. Crunches like an apple but tastes like a grape. Ayan, grapple. The second example is a type of cow that produces milk that contain human protein and found to be more nutritionally balanced. So it's a type of cow called the transgenic cow. So yung mga sumasagot na modules ha. So number 1, grapple. Ang tawag natin sa number 2 is your transgenic cow. Number 3 is a type of cabbage. Ito type of cabbage that is injected with scorpion venom aimed to kill caterpillars. But take note yung, of course, yung they modified the venom sa scorpion. Ang target niya to kill the caterpillars, not the consumers, not the human consumers. Okay? So ang tawag dyan ay venomous cabbage. Okay? That's number 3. Number 4 naman, they can produce virus proteins against hepatitis and cholera. Ayan, sa banana. Okay, it's called the banana vaccine. So they're aiming or studying to propagate bananas that can contain or they can produce virus proteins against hepatitis and cholera. So sa dalawa sa mga common na diseases all throughout the world, di ba? Pero hindi siya yung technically na yung banana talaga na ganyan, kakainin mo siyang ganyan. It's actually, ipaprocess pa siya para maging oral vaccine siya. Para siyang yung kinakain ng mga babies na banana, imamash. Ayan. So ipaprocess pa siya para ma-consider siya or mag-work siya sa katawan pag kinain as a vaccine. Hindi siya yung totally na yung saging, you will eat it, may vaccinated. You are vaccinated already. So it will underwent a process pa. So banana, bakit banana yung sabi sa studies? So based on my research, so madali lang kasi mag-reproduce ang banana. It's one of the staple food din sa different parts of the world. Okay? So that is our banana vaccine, number 4. Number 5, insulin gene is injected to this bacteria. So bacteria, anong klaseng bacteria kaya yan? To produce the hormone insulin. Ayan, para sa mga diabetic. Ayan, so diabetes is one of the critical illness na mayroon sa buong mundo, di ba? So we have, or they are utilizing the Escherichia coli or the E. coli bacteria to produce the hormone insulin. Number 6, ayan, would you imagine this? Would you believe that this actually exists? Spider, the silk yung silk na galing sa spider, ayan, incorporated sa genes ng goat, ayan, spider enhanced milk that can produce silk stronger than steel. Ayan, so they find other means para makapag-produce pa ng silk. Kasi ang silk, silk moth lang ata yung isa sa mga source ng silk, di ba? So they tried to investigate ano mangyayari kapag in-incorporate mo yung genes ng mga silk spider sa goat. Ayan, so you have the product called the spider goat. Alright? Ayan, tama. Spider goat, sir. Thank you so much. Ayan, number 7, a cat that produces a fluorescent protein in its fur. Ayan. So basically, ginagamit to for advanced research. Ang tawag natin dyan is the luminous cat. And finally, na-mention ko na ito kanina, a hybrid of the tiger and the lion that will end our activity for module 25, page 11. Of course, we have the liger. Ayan. So we're done with with that activity. We are now ready to discuss the different benefits of genetic engineering. Ayan. So ano nga ba yung mga benefits? So kanina sa beginning ng session natin, sabi ko, sana ma-highlight yung benefits. Kasi naniniwala ko na yung science, when applied, or when applied, or technology applied, na kapag maganda intention mo for the benefit of humanity, syempre, dapat suportahan yung mga ganyang klase ng studies. Alright? So one of the benefits of genetic engineering, of course, is the improvement of the quality and crop yield. So we have several crops na products na actually ng GMO. Ang goal naman kasi, let's say for example, the tomato, madaling mabulok kasi ang tomato, di ba? So because of genetic engineering, we can produce tomatoes that are large, mas malaki, so for food security purposes. And number 2, hindi sila madaling mag-spoil or yung shelf life nila matagal. Okay, so that's one benefit. The second is, because of genetic engineering, we are able to produce drugs to treat different kinds of diseases. And not only to treat, to also diagnose different diseases like HIV, tuberculosis, hepatitis, allergies. And meron din tayong tinatawag na konsepto, the term is gene therapy. So gumagamit ng healthy genes na transfer sa gene ng tao may sakit para to cure different diseases or para ma-improve although not to cure but to improve. Kumaga nasa clinical trials pa lang kasi. Just like based on my research, we have cancer, ayan, hemophilia, yung hindi nagka-clot yung blood, di ba? And many others. Ayan, so that's the second benefit of genetic engineering, production of drugs to treat diseases. We also have you heard of biofertilizers and biopesticides? Kasi tayong mga tao, di ba, sa mga plantito tsaka plantita or mga nasa farming industries, we incorporate or we add fertilizers or pesticides, pwedeng natural yan or chemically produced. But because of genetic engineering, we are using microorganisms that will serve as natural source of pesticides. So imagine that. So may mga bacteria pala na ginagamit or in-engineer to produce allelopathic genes that will serve as natural pesticides. For fertilizers naman, GMOs are grown symbiotically with plant. Say for example, di ba, nitrogen is very important sa mga halaman. Ayan. So nitrogen, it exists, so nitrogen cycle, no, kailangan ng nitrogen-fixing bacteria. But because of genetic engineering techniques, say for example sa mga soybeans, they were able to produce, yung plants na yun, they were able to produce their own usable form of nitrogen. So hindi naaasa talaga sa, oh yes, we have the natural nitrogen cycle, but because of genetic engineering, makaka-incorporate na symbiotically ng nitrogen as natural fertilizer. So, di ba? Very advantageous. And then finally, have you heard of bioremediation? Ayan, para naman to solve environmental problems. Ayan. So, bioremediation, to give you an overview or an idea, ayan, we have here a diagram. So, ang ginagawa natin for bioremediation, it uses natural and recombinant microorganisms to break down toxic and hazardous substances in the environment. So, class, naturally, meron tayong syempre, of course, bacteria and other microorganisms that naturally break down yung mga basura, mga substances na hindi natin kailangan para ma-recycle siya in nature. But because of this technique called bioremediation, ginagamit or mas na-enhance pa yung mga microorganisms na to in terms of speed at saka efficacy sa pag-degrade nila ng waste materials. And also, have you heard of bacteria? So may nakapag-develop na rin or nakapag-engineer na mga bacteria that is called the oil-eating bacteria. So ito yung ginagamit naman pag may oil spills, di ba? Nagkaroon ng mga leaks sa mga ano natin, mga planta natin, di ba? So yun, imagine, very small organisms but very huge help sa ating environment. So biofertilizer, biopesticide, at saka bioremediation. So Sir Gallardo, yeah, the flavor sour tomato, ayan, sa mga unang genetically modified organism. Thank you for sharing, sir. Ayan. So for our last test this afternoon, all we have to do is classify lang natin kung benefit or risk. So B for benefit and then R for risk is found on Module 25, page 14 of your module. Ayan. So number 1, obviously crops like potato, tomato, soybean, and rice are currently being genetically engineered to obtain new strains with better nutritional qualities and increased yield. So para malabanan yung microdeficiency, malnutrition, tsaka magkaroon ng food security. So ginagamit natin yan. Of course, that's a benefit. 1 is B. Number 2, genetic engineering in food can be used to produce totally the same or identical substances such as proteins and other food nutrients. So in terms of nutrients na naman, so we have B. We have benefit, of course. Number 3, positive genetic engineering deals with enhancing the positive traits. May clue ka na, ang dami positive. Positive traits in an individual like increasing longevity or human capacity, while negative genetic engineering deals with suppression of negative traits in human beings like certain genetic diseases. So pinapabagal or pinapa-enhance pa ang quality ng life natin. So that's of course a benefit. Number 4, hindi ko mabula sa comment section natin. Number 4, genetic engineering in food involves the alteration of genes in crops. That's a risk. Kasi minimodify natin yung natural, yun nga, yung sinasabi, mamamodify natin yung natural na yung takbo ng ecosystem. Alright? So that's a risk. Hindi natin maiwasan, well, but that's one factor that we should consider. Okay? Number 5, while increasing the immunity to diseases in plants, the resistance genes may get transferred to harmful pathogens. Okay, so that's another risk. Number 6, genetic a risk. So negatively naapektuhan niya ang diversity ng organisms natin in the wild. And number 7, the genetic modification of foods can be used to increase their medicinal value, thus making edible vaccines available. Yung kanina, di ba, yung sa banana vaccine. That's a benefit. Okay, so those are the answers for Module 25, page 14. So I hope na-enlighten kayo sa konsepto ng genetic engineering at mas na-highlight, of course, yung benefits ng technique na ito para ma-improve yung quality ng life ng ating mga tao. And of course, as scientists, kayo, mga learners natin, as future geneticists, as future molecular biologists, or other scientists in the field. So sana mas lamang yung benefits, di ba, na sa future innovations na gagawin ninyo. Alright? So consider natin yung the benefits rather than of course, we have to consider the risk factors. We have to balance it out. Okay? Ayan. So for our hashtag Be Inspired, hashtag Be Blessed segments, yung mga last segment natin during our session, according to Darren Hardy, so I quote, DNA has nothing to do with success. Sabi niya. So kahit ano pang ganda ng DNA mo, matalino ka, you have pleasing personality and all. Ayan. Turn your genes into overalls and get to work. So, ibig sabihin lang dyan ni Darren Hardy, you have to maximize kung ano yung binigay sa'yo ni God, di ba? Binigyan ka na ng talino, binigyan ka ng specific trait, magandang trait, gamitin mo yan para ma-bless mo yung sarili mo, yung family mo, at syempre, para makatulong ka sa pag-unlad ng ating bansa in the future. Alright? So, that would be all for our session this quarter 4, week number 4 of Senior High School Earth and Life Science. You can communicate me via email, Facebook. You can check my Week4 account and my YouTube account, Mr. Voice Educator PH. I hope you had fun sa ating 40 minutes plus na discussion. Ayan. Thank you very much. See you again next week for our session, Senior High School Earth and Life Science. This has been Sir Tony. Ingat po tayo palagi and God bless. Bye-bye. Thank you so much po.
Sigurado ako na marami ka na namang natutuhan sa ating ETUlay tutorial session ngayong araw. Tandaan, ito ay hindi lamang para sa ating mga mag-aaral, kundi pati rin sa ating mga minamahal na guro at mga magulang na kaagapay natin para maituloy ang pagkatuto sa kabila ng nararanasang pandemya. Patuloy ding sumubaybay sa DepEd TV para sa mga araling ginawang video episodes. Mapapanood ito mula Lunes hanggang Sabado, alas 7 ng umaga hanggang alas 7 ng gabi sa inyong mga telebisyon. Abangan bukas mula 1:00 ng tanghali ang iba pang aralin sa ating ETUlay free online tutorial session sa English. I-like and subscribe at manatiling nakasubaybay sa ating ETUlay tutorial session sa DepEd Edtech Unit FB page at Educational Technology Unit channel sa YouTube at sa DepEd Tayo at DepEd Philippines social media accounts. Paalam!